preparation of dilutions of hydrogen peroxide. The hydrogen peroxide is a 30% solution, so we will be making serial dilutions using these 50 milliliter tubes initially, and these can serve as a source for aliquots that can be distributed to students. I'm going to begin by pipetting 9 milliliters to each of these tubes. Hydrogen peroxide is temperature and oxygen sensitive, so we want to mix these thoroughly but gently to avoid deactivating the hydrogen peroxide, and we keep it on everything on ice. And the vehicle that what we're diluting with is a solution of 2/10 TE. It contains 2 millimolar tris, pH 8, and 2/10 millimolar EDTA, an agent that chelates metal ions, and thereby protects the hydrogen peroxide. And then for the serial dilution, 1/9 of the volume of 1/9 of 9 milliliters is 1 milliliter. So that's what I'll be using to transfer in making the serial dilution. Add that to the tube called 3% hydrogen peroxide. Mix by pipetting up and down. And I'm also going to mix just by gently inverting the tube a couple of times. And now I'm ready to transfer the next milliliter. Both of these tubes get capped. And then these are distributed to the students as smaller aliquots. Each student will need 100 microliters, so it would be sufficient to give them 200 or 300 microliters in little Eppendorf tubes. Mix them one more time before distributing. In this third tube, got the vehicle, the 2/10 TE, and I labeled it no stress, 0 hydrogen peroxide. And just to keep everything uniform, give that a good mix, even though it's not really a mixture at all. So I'm just going to be distributing these solutions in smaller aliquots best to prepare these the day of the lab. The shelf life is unknown. So here I'm going to label this 0 HP, 0 HP, 0 HP. And then the 3% HP, 3% HP. And then the 0.3%. And this is the solution current, the current solution concentration of hydrogen peroxide. Once the students prepare the reactions, the hydrogen peroxide concentration is decreased by a factor of 10 one more time. So 100 microliters of this added to the cells will create the 0.3% hydrogen peroxide condition. 100 microliters of this added to 900 microliters of cells will create the 0.03% hydrogen peroxide. And of course, if there's no hydrogen peroxide, this will be the no stress control. I'm going to choose 300 microliters. And for this work, you don't need to change tips. If you go from decreasing concentration to increasing concentration. And I chose 300 microliters just in case somebody needs to start over. View. That was the 0.3% hydrogen peroxide. It's always a good idea to check your labels before and after you start distributing a solution, that you can catch errors before they cause problems. This is the 0.3% tubes. One final set to distribute. 3% hydrogen peroxide. And then I will be returning this to the cold cabinet, and also these solutions are no longer needed. They might be useful for the next group, and so I'll put them in the cold cabinet. There's how you make the hydrogen peroxide solutions.